Hello everyone, thanks for tuning in to my infographic presentation. My name is Beth Rasmussen and I'm a second year master's student in the Avian program. So my infographic describes some of the research I've been doing in the past few years and what will be one chapter of my thesis. First, I'd like to direct you to the project basics section. So between 2016 and 2018, our team tested 1,344 ring-billed gulls and 244 Franklin's gulls for avian influenza virus, which will be referred to as AIV throughout the presentation. So our goals were to see which species had a higher AIV prevalence and if either species harbored an H5 subtype. And when I say H5, this refers to one of the proteins on the surface of AIV that is used to characterize subtypes. So the reason we're concerned whether or not Gulls have H5s is because they have the ability to change into highly pathogenic viruses. Highly pathogenic viruses are extremely deadly to domestic poultry. So if we move over to the Y box, in 2015, the US poultry industry experienced an outbreak of highly pathogenic H5 AIV. And our main goal was to see if gulls played a role in this outbreak. And the image on the left is a ring-billed gull, which was one of our study species, and it is shedding AIV from its mouth and cloaca. And next, I'd like to direct you to the how section. So we captured gulls using either a net launcher or by hand nets, and one not so scientific finding is that gulls can be lured in with popcorn. And this image shows the two swabs we took from each bird, one from the mouth and one from the cloaca. And when these samples got to the laboratory, we used a method called RT-PCR, which tests for the presence of viral particles. So if the sample was positive for AIV, we then further tested it specifically for the H5 subtype. And below this are some selected results. So we found that ring-billed gulls had higher AIV prevalence than Franklin's gulls. For oral swab, 65% prevalence in ring-billed gulls versus 35.8% prevalence in Franklin's gulls. And for cloacal swabs, ring-billed gulls, 21.5% prevalence versus Franklin's gulls, 17.7% prevalence. And we also found no H5 subtypes in any of the samples tested. And next, I'd like to direct you to the right side of the infographic in the significance box. So during the 2015 outbreak, 9 million turkeys were either directly killed by the virus or had to be depopulated to contain the spread of the outbreak. And this is over 20% of all domestic turkeys in Minnesota, according to USDA APHIS and the Minnesota Turkey Growers Association. And unfortunately, the source of the outbreak is yet unknown, but we did not find any highly pathogenic viruses in any of our sampled gulls. And finally, one of our next steps is going to be looking at the viral genetics of some of our viruses to see where they originated from geographically. So thank you so much for listening. I'd like to thank my funders, the Minnesota Agricultural Experiment Station, and the Environment and Natural Resources Trust Fund, and finally my advisor, Dr. Marie Colhane. Thank you.